As we look at T cell development, we're going to break it up into two phases. Much like what we did with B cell development, we broke it up into phases and talked about what happened at each one. We're going to do the same here. It's just that there's not as many phases, or at least we're just focusing on the first half, and we're not looking at the other phases of when a T cell is mature and goes out and looks for infection. So phase one is all about becoming a T cell, and a way a cell becomes a T cell is it puts out a T cell receptor along with a co-receptor, and that's what makes it a T cell. So we're going to look at what it takes for a T cell to become, or to go from being a non-committed T cell, not committed like lymphoid lineage, into being either a CD8 or a CD4 positive T cell. And again, this all happens in the thymus. So the bone marrow will produce a progenitor cell. It's lymphoid lineage. It might be a B cell, might be a T cell. It's not committed. But those that go to the thymus then are committed then to becoming T cells. And at this point, they're going to have CD34 on their surface, but no other distinctive markings. So nothing that says it's going to be a T cell, nothing that says it's going to be CD8 or CD4, nothing like that. Their T cell receptor genes are still in germline configuration. There's been no somatic recombination that has occurred yet. All of this will be initiated once those progenitor cells start to interact with the thymic stromal cells. That's when they're signaled to differentiate, commit to becoming a T cell, and start to express or rearrange the genes to express the T cell receptor. A critical cytokine that drives this differentiation is interleukin-7. And so interleukin-7 is secreted by these stromal epithelial cells that tells a non-committed progenitor cell, you are going to be a T cell. And then the progenitor cell's like, okay, I guess I'll be a T cell. And so then that's when somatic recombination will start to occur. Another major regulator of T cell development is something called Notch1. Notch1 is a protein that is a surface receptor on the thymocytes, so on those progenitor cells, that will interact with the thymic epithelial cells. And together, these interactions that the progenitor cell has with the thymic epithelial cells and the secretion of the interleukin-7 to the progenitor cell will drive that cell down the T cell pathway of differentiation. So here's a side-by-side comparison of what a progenitor cell looks like, an uncommitted progenitor cell, to a immature T cell. And you can see that CD34 is going to be found on the progenitor cell, but it will not be found on the T cell as committed to, or the cell that is committed to the T cell lineage. Now, at this point, though, once these cells interact with the thymic epithelial cells and get the signal to move down the T cell pathway of development, they're not going to have CD4 on the surface or CD8 yet. And so what we do is we call these cells double negative because they don't have those co-receptors on there. They will have a variety of other markers on their surface, but they won't have CD4 or CD8. Those do not exist yet. And so that's why they're called double negative T cells. But they do have the reg complex that is needed for somatic recombination. And so the reg complex, those are the genes that will create the reg proteins that will turn on somatic recombination, recognize those switch sequences and bring in the different V's and D's and J's. So let's then go ahead and take a look at the steps that a double negative T cell will go through to become an immature T cell. We're going to look at this image on several slides. And every time we look at it, I'm going to be focusing on a different area of the image. And I'll have it indicated by a red arrow, but I'll also try to make some markings on it as well. So recall when we talked about T cells in or T cell receptors in chapter 5, we said that there's two chains, right? There's an alpha beta that can be paired together, and then there's gamma delta. And so you can have an alpha beta T cell, you can have a gamma delta T cell, and those are based on the types of chains that make up the T cell receptor. So the process then of T cell development could give you either or. Okay, once development starts, it could go down the alpha beta line, it could go down the gamma delta line. And how it happens is really a series of races that occur, because they both of these cell types develop from that same double negative progenitor cell, that uncommitted or that committed T cell, but it's double negative. And what will happen is the first thing is the beta chain and the gamma and the delta chains are going to start to go through somatic recombination. Let's look at what that looks like just to remind ourselves, because it's been a while. So here is our diagram that we can pull from chapter 5, where looking at the alpha beta chains, you could also substitute these alpha and betas for gammas and deltas, that's fine. But with the beta chain being the first one going through somatic recombination, a J and a D segments get paired together, and then a V segment is brought in, and you get your D to J, V to D to J rearrangements. Okay, that happens, and then a constant region is brought in. Okay, one of the many constant regions to get a beta chain. Okay, this can happen at one locus. Actually, this can happen at both loci. Okay, so you have two you have two homologous chromosomes with the beta chain gene on it, and this rearrangement is happening on either one. Okay, and just like what we saw with B cells, there can be productive rearrangements and there can be non-productive rearrangements. So then when we look at the alpha chain, we don't have the diversity segments, but rather we just have one of these J segments being brought in, one of these V segments brought in, and then the constant region is brought together. And so that'll happen in a little bit, but right now we're just focused on the beta chain rearrangement, okay? So this is happening at the same time on both chromosomes, but at the same time, the gamma and delta chains are rearranging as well. And so you have the possibility, so there's this race. You have the possibility that a beta chain can be produced, a productive rearrangement, or you have the possibility that a gamma and a delta chain can produce a functional rearrangement. If it's a functional gamma delta rearrangement first, then that T cell is going to commit to becoming a gamma delta T cell. And so you see that happen. That's if that occurs first. If a beta chain, though, just a single beta chain is produced and it's a functional product before the gamma and delta chains, then the T cell is like, oh, I have a beta chain. Let's go ahead and stop rearrangement, because this one seems to work. And let's go ahead and decide to rearrange the alpha chain. But before that, let's go ahead and make what we call a pre-B cell receptor. We'll take a look at that in a little bit. But this beta chain hangs out in there. And the cell now has decided to commit to putting out CD8 and CD4 on its surface. Now it's called a double positive T cell. So if the beta chain makes a productive rearrangement before the gamma and delta chains are able to make a productive rearrangement, the cell will go ahead and make what's known as a pre-B cell receptor, but also produce both CD8 and CD4 and become a double positive T cell. Just like what we saw with B cells, when the heavy chain was made, proliferation occurred and a whole bunch of cells were made. This also is the case when a beta cell or when a beta chain is made, that a bunch of cells will be made. So you have like 100 or so of these double positive T cells that have the same beta chain, but now could potentially make a different antigen specificity by making a variety of different alpha chains. Just hold on to that. Okay, so if the beta chain is produced first and beats the gamma delta chain rearrangement, the cell commits to become, well, it doesn't commit, but it does commit to making CD4 and CD8, putting it on the surface, and then starts to rearrange the alpha chain. But now, if alpha chain starts to rearrange, that means the reg enzymes are turned on again. And that also means then not only is alpha being rearranged, but so are gamma and delta again. So now they get to come back in and they try to race alpha chain. So it's more competition. You have a double positive T cell that is going to be making, or at least trying to make, an alpha beta receptor before gamma delta chains can rearrange. Or you have a cell that could make gamma delta chains and commit to becoming a gamma delta T cell if it ends up making those before the alpha chain rearrangements. So cells that fail to make productive T cell receptor rearrangements die by apoptosis, whether it's gamma delta or alpha beta. And that's where those thymic macrophages are so important, because apoptosis is the fate of almost all T cells that go on through the development process. It's crazy. Only about 2% of T cells are actually able to make productive alpha beta or gamma delta rearrangements. It's just mind-blowing how many cells don't end up making it. Okay, so let's now go down the path of gamma delta. So we're going to kind of take a step back and build in some knowledge that I kind of just glossed over. If a thymocyte that is now doing this race between gamma delta and beta, if this thymocyte makes a productive gamma and delta gene rearrangements, and they're productive, and they're able to do that before beta chain is able to create a productive rearrangement, the cell will become a gamma delta T cell. And so we call it gamma delta T cell. I'll put the little colons in there. Okay. That is the way that gamma delta T cells are formed. When this happens, the gamma delta T cell heterodimer is moved to the surface of the T cell, and that's going to stop reg enzymes from recombining or rearranging the beta chain. And then none of this happens down here because we have a committed gamma delta T cell. But more common is that a beta chain is going to be produced before both a gamma and a delta. So actually, this doesn't happen over here very often. Like this is like not very often, because two chains have to make productive arrangements versus just one chain. So more often than not, a beta chain productive rearrangement will occur first, or will win the race. We have two options. The cell can try one homolog or try another homolog, because there's two chromosomes for the beta chain genes. And so, okay, so the potential then for trying up to 4 beta chains, because we have homologous chromosomes and the second rearrangement on the same locus, you have 4 chances that can actually produce a productive beta rearrangement. And that means that that race happens more often, or the race ends up with beta winning more often. So T cell lineage then is going to have a bias in the favor of alpha beta, because only one beta chain has to be produced where to get a gamma delta T cell, both gamma and delta chains have to produce productive rearrangements before one. And so it's just sheer numbers. 2 takes longer to get than it would take for 1. So more often than not, you see the cell produce a beta chain, make a pre-B cell receptor, and then put out CD4 and CD8 on their surface. Now, what is this pre-B cell receptor? Well, just like what we saw with, or not pre-B cell receptor, pre-T cell receptor, sorry guys. Just like what we saw with the B cells, beta chains are made and then tested for their ability to potentially bind with an alpha chain, with a surrogate alpha chain. And this surrogate alpha chain is called PT alpha. PT alpha is invariant. So PT alpha is the same. It's just a regular gene, transcribed, translated, to make an invariant protein chain that will associate with a beta chain. If it does, then the beta chain seems like, okay, this is probably a pretty good chain. It's probably good chances it's going to bind with an alpha. Sounds good. And that's what that pre-T cell receptor is. So if the beta chain binds pre-T alpha, that will then assemble with our CD3 complex. Remember, because the CD3 complex is also needed in association with the alpha and beta chain to make the full T cell receptor, and puts it all out onto the surface of the T cell. The deal is that it's on the T cell, but there's no antigen specificity because those spots are blocked by the way that pre-T alpha interacts with the beta chain. So that's why it's called a pre-T cell receptor. And this is then going to be that first checkpoint to make sure that a beta chain is going to, well, that a T cell receptor is going to be functional. Okay. Let's then talk about, just kind of review the 3 possible outcomes that we have so far. Number 1, when a beta, okay, just so far with the beta and the gamma delta race. First of all, a gamma and a delta chain productive rearrangement can be made. And that means that the T cell will commit to becoming a gamma delta T cell. Okay. More common though, the beta chain is going to rearrange first and then interact with pre-T alpha, creating a pre-T cell receptor that's going to then go back and start to rearrange alpha. But most common, none of this happens and the cells die by apoptosis in the thymus, cleaned out by the macrophages. So, but really, like, we're just going to be focusing on either 1 or 2. Okay. When that successful beta chain is rearranged, because that's going to be the more common situation, that stops reg production, which shuts down not only gamma delta chain rearrangement, because they lost this race, but also shuts down beta chain rearrangement because there is a functional beta chain now. And so the cell will stop rearrangement, commit to that one beta chain, and that is another example of how allelic exclusion occurs, this time with T cells. So there won't be different T cell receptors on a given T cell, they'll all be the same. Well, they'll all have the same beta chain, for example. Okay. You'll see what I'm talking about in the next lecture. T cells are going to proliferate at this stage and make a lot of these cells that have identical beta chains. And they're also going to produce CD4 and CD8 and put that up on the surface so that they're called double positive T cells. They're still immature, but now they're double positive. And the reg complexes are turned on again at this point to start the race again, but this time the race is going to occur between alpha and gamma delta. Here's our favorite picture again. Now we're looking down here where we have alpha rearranging as well as gamma delta rearranging at the same time. So it's another race, and there can be repeated attempts of the alpha chain gene rearrangement. when a double positive T cell, because now we have our CD4 and CD8, our double positive T cell makes a productive alpha chain rearrangement, then it's going to be tested, it's going to be transcribed, translated, and it's going to be tested for its ability to bind with the existing beta chain. That's the second checkpoint of T cell development. If that alpha chain is able to bind to the beta chain, that double positive T cell is selected to survive and go on to further testing or go on through further development. But if the alpha chain does not assemble with the existing beta chain, it can try again. Now, gamma and delta is still trying to rearrange, but if alpha is able to eventually find a combination of segments that makes a chain that is able to associate with beta, then the T cell will be an alpha beta T cell. But at any point, if gamma delta produces a rearrangement first before alpha can make a functional chain, then we will get a gamma delta T cell instead, even though beta chain is hanging out. The beta chain goes away, the CD4 and CD8 go away, and the cell just ends up becoming a gamma delta T cell. But that's not very common because, again, there's two chains that have to rearrange versus just the one alpha. So more common, the alpha chain is going to be able to rearrange and make a productive rearrangement first, join with the beta chain and form a alpha beta T cell. So this is going to be more common, and that's why the alpha beta T cell is our default T cell, because there's just many more of them. And third, of course, the option could be where alpha is not able to produce a productive rearrangement, neither are gamma delta, and so then the cell dies by apoptosis in the thymus. So the outcome for this first part then of development is getting a set of rearranged T cell receptor genes that are able to associate with each other and be produced out onto the T cell. If it's alpha beta, then there's also going to be the production of CD4 and CD8 on the surface, and both of them at this point. Again, we have our double negative when you don't have CD4 or CD8, and that's when gamma delta and beta are rearranging, and then double positive where gamma delta and alpha are rearranging. But we're just kind of looking here at alpha beta progression. And so we have our checkpoints along the way, just like we did with B cells, and then eventually we end up with a committed alpha beta T cell, but it's double positive. So here is a diagram that does a summary of these checkpoints, and it also combines the path that the thymocyte or the developing T cell is taking through the thymus, where it enters kind of at this medullary cortex medullary zone, will move up through the cortex and then back down toward the medulla as it is rearranging its genes. We start out with a double negative cell rearranging the alpha or the gamma, delta, and beta. Checkpoint, we're just looking at alpha beta, that makes it a lot easier. Checkpoint to make sure if the beta chain rearranges that it can associate with a surrogate alpha chain. And then we have our double positive T cells that are rearranging the alpha chain. And the second checkpoint then is to make sure that the alpha chain does in fact associate with the beta chain. And then you have mature double positive T cells at this point. All right, we'll end there and we'll pick up the last part of development in the next lecture.